So today I'm going to talk to you about CloudScreen, a graph-based drug repurposing platform empowered by machine learning. I would like to start by presenting some general information about our platform and our machine learning approach, and afterwards my colleague Sotiris will demonstrate some code and queries for each step of the pipeline. And then, if the time allows, we can take some questions and discuss. But first, what is drug repurposing? Some of you may know that drugs work by physically interacting with and altering the function of specific proteins in our body. Specific proteins are linked to diseases and their outcome. In the human body, there are approximately 20,000 proteins consisting the entire proteome, from which only a fraction is known in terms of structure. So basically, drug repurposing is the process of uncovering new target proteins or indications of approved or abandoned drugs for use in a different disease. When it comes to drug repurposing compared to traditional drug discovery, the comparison says a lot. Repurposing drugs have better success rates, less safety concerns, fewer years to impact, and much fewer costs. So, as you can imagine, it's a really important technique in the field of drug discovery. CloudScreen is a graph-based platform developed with Neo4j by Cloudfarm Private Company and the Biomarker Discovery and Translational Research Laboratory of National Hellenic Research Foundation. CloudScreen integrates two types of data, 1D biomedical data and 3D structural data, and feeds them to an AI predictive algorithm in order to produce effectiveness and toxicity predictions, which will be experimentally evaluated, leading to new indications. Now, the field of drug repurposing has several limitations on how it has been approached. Most solutions are heavily based on textual data, while we approach the problem by incorporating 3D structural protein information, focusing at the molecular level. Existing protein structural information account only for 20% of the proteome, while we are curating and implementing 100% of the proteome space. Also, there is a vast amount of contradictory information in existing scientific data. We are trying to tackle this limitation by our own machine learning algorithms. 1D biomedical data in CloudScreen concern information extracted from track patents by applying name entity recognition, data collected from clinical trials, the clinical significance of protein variants and their disease associations, pathways related either with a protein or a drug, pharmacogenomics information, data related to pharmacovigilance, information about protein disease associations, and drug activity from experimental data. 3D structural data include the structure of approved and experimental drugs, the experimentally known structures of human proteins, homology models of missense mutations of proteins forming the human variome, AlphaFold models with high confidence allowing us to explore the human protein, and molecular docking calculations of the approved and experimental drugs upon the proteome and the variome, allowing us to map the interactions between drugs and proteins. Molecular docking is a molecular modeling method which predicts the preferred orientation of one molecule, one drug for example, to a protein's binding cavity. The 1D and 3D data have been integrated in a graph database employing Neo4j. The core of CloudScreen is the 1D data which form a knowledge graph. The core knowledge graph is further extended by the addition of the structural 3D data extracted from visual screening. The knowledge graph has been designed by integrating data from public repositories, while the 3D data come from in-house calculations performed in a high-performance computer. The graph database was selected since it was the best way to represent the complex interaction of biochemical data. Yet, storing all this information in graph format has also assisted in better visualization and analysis of the data. It is important to mention that during the development phase of the CloudScreen platform, we found that the graph database is easily expandable. Nowadays, artificial intelligence constitutes a crucial part of the data analysis routines. We can define artificial intelligence as the development of systems and machines that carry out tasks which typically require human intelligence. In data science, where data is transformed to information, machine learning, which is a branch of artificial intelligence, is applied in order to produce accurate predictions that can guide decision-making without human intervention. Machine learning utilizes that data to recognize and learn hidden patterns which cannot be easily detected by humans. To this end, the graph-based machine learning is used to process network data, leveraging the power of the relation between entities that can be used for predictive, modeling, and analytics tasks. The main challenge in graph machine learning is the transformation of the network to a tabular data, which is usually done through node embeddings. Graph machine learning can have many applications, with the most common ones being node classification, link prediction, community detection, and node regression. CloudScreen utilizes graph machine learning as an extra layer of validity to the predictions that it provides. More specifically, the outcomes of the main queries that the platform has are further combined and filtered through the graph machine learning predictions. The purpose of CloudScreen is to provide suggestions about drug repurposing candidates. This is done either through a drug query, where the platform provides the new targets for existing drugs, or through a protein query, which results in drugs that possibly target the queried protein. The results of those queries are mainly guided by molecular docking results. So the graph-based machine learning is used complementary to the docking calculations. These two technologies combine the molecular and topological features to provide and integrate highly accurate prediction. CloudScreen applies graph machine learning for link prediction in order to identify probable drug-protein interactions, drug variant interactions, and drug disease interactions. The steps for the proposed graph-based machine learning pipeline are the following. At first, we need to have set up a knowledge graph. In our case, it has the known drug-protein interactions and experimental data for the activity of a drug to a protein target. Then, the features of the graph should be extracted to quantify the topological drug-protein associations. So, feature engineering is applied by implementing node embeddings to transform nodes to vectors. Since the nodes are either drugs or proteins, in order to quantify the interaction, the vectors produced by each node type should be combined. The feature combination is done by geometric approaches that will be explained later. Having the feature vectors for each drug-protein interaction, a matrix is designed, which is preprocessed before machine learning. The main preprocessing steps are duplicate removal, data normalization, splitting the data into classes, and then dividing the data into training and test sets. Then the training of the selected machine learning algorithms is performed, and the trained models are validated upon an external holdout test set. The test set is used to evaluate the model's performance and generalization capacity. Having an accurate and robust model, we can perform predictions in a set of drugs and proteins of interest. Finally, the resulted predictions should be further analyzed to identify the most probably repurposed drugs. So, for the feature extraction, we perform node embedding techniques. Node embedding is the process of transforming a node into a vector. Currently, Neo4j supports three types of node embeddings. FastRP is a random linear projection algorithm which projects n number of vectors of arbitrary dimension into all number of dimensions and still approximately preserve pairwise distances among the points. GraphSage is an inductive algorithm which utilizes node feature information to generate node embeddings by sampling and aggregating features from a node's local neighborhood. Node2Vec computes a vector representation of a node by sampling a neighborhood through random walks in the graph. Then the algorithm utilizes these random neighborhood samples to train a single hidden layer neural network to predict the likelihood that a node will occur in a walk, based on the occurrence of another node. For the feature combination, several techniques can be used. Some of them are vector concatenation, vectors Hadamard product, weighted L1 normalization, weighted L2 normalization, and cosine distance. The calculation of each technique is shown in the results column here. For the training and the validation of the model, the initial dataset, which has both positive and negative classes, is splitting in training data, which consists the 70% of the dataset, and test data, which represent the 30% of the dataset. The model is trained by tenfold cross-validation and applied in the training dataset. And then the resulting model is evaluated against the holdout test set. So in a few, the final model can now be used for the link prediction in unknown drug protein pairs. The prediction of a new link will indicate a new target for an existing drug, enable drug repurposing. The new link predictions should always be experimentally evaluated. So, in a few words, using Neo4j, we accomplished to integrate 1D and 3D biochemical data inside a knowledge graph and then combine the docking results with graph machine learning in order to develop a drug repurposing platform for intellectual property generation. And now it's time to hand you over to my colleague Sotiris for the demonstrating part of our session. Thank you very much.
So, hello everyone. Now we're gonna proceed with the demonstrative part of the session. Let me share my screen. So, you can probably now see my screen. We will begin with a quick overview of our platform and some statistics. So, as we can see from this query, we have approximately 300,000 nodes in our graph, whereas we have approximately 19 million edges, which represent the relationship among our nodes. More specifically, we can see in the above query that we have 33 type of nodes, which represent 33 different heterogeneous open repositories from which we have collected our data. So we have 92,275 proteins in our platform and 14,316 drugs. So we can imagine that the amount of data is very large, and the docking experiments that were done in all this amount of proteins and drugs resulted in some 100,000 of dockings. So we can see here the number of nodes for each type of database, the average number of properties per node, the minimum number of properties per node, the maximum number of properties per node, and the equivalent statistics for the relationships. Now we're gonna proceed in the graph machine learning part. But at first we have to understand the type of data that we have aggregated in order to perform the graph machine learning. So I will begin with a query that returns the drug protein pairs, which will be used for the positive class. As a positive class in our graph machine learning pipeline, we mean the pairs of the marketed drugs and proteins. So in this case, we can see that we have subgraphs that contain a blue node, which represents the drug, and a red node, which represents the protein. We can see the connections among drugs and proteins, which are the known connections. Its drug node, its blue node, has some properties. If the drug has been approved or not in a specific protein target, which is used in our pipeline in order to collect only the approved drugs for specific protein targets. The second part of the knowledge graph that we are using in our machine learning pipeline is the data for the negative class. As negative class, we define the protein drug pairs that are not successful. These data are mainly retrieved through experimental evaluation from public repositories. So here we can see again the blue node is a drug, whereas the red nodes are again proteins, but this time the edge that connects the two nodes carries the information about the experimental evaluation of the drug in the specific protein target. So we have used a cutoff, which will be shown later on, in order to define only the failed experiments in order to represent the negative data. So, excuse me. All right. What we have performed in our pipeline is the integration of the Neo4j platform with VR Studio. We have specifically connect RStudio with Neo4j in order to retrieve the data in R and perform the analysis in the RStudio. So the connection is done through this chunk of code where we use the Neo4R driver, and it is the first step of our pipeline. We have to connect to the knowledge graph database in order to retrieve the data. The second step is to query the drug protein pairs, which are about the positive class and contain the known pairs of marketed drugs in specific proteins, whereas the third step is projecting the above graph in the graph catalog. This projection step is very critical to be performed since we use the graph data science library of Neo4j, and we should perform this projection in order to apply the node embedding algorithm later on our pipeline. So the node embedding algorithm that we used in our case is FastRP. We have also implemented node2vec, and we will provide the results later on. So in this way, we perform the FastRP query in order to generate the node embeddings and transform our nodes into vectors. The fifth step of our pipeline is now to combine the vectors that we have produced. We should take into consideration that each node of the graph is described as a vector, so we have no connections, we have no data about the connections of drug and the protein. So by performing Hadamard dot product, we combine the node vectors from a drug with the node vectors from a protein, getting the features that describe the relation of the two nodes. The sixth step is querying the experimental data in order to create the negative class. In this case, as you can see here, we have put a threshold which refers to 10,000 nanomolar, which is a metric used in the experimental data, in every type of experimental data, and the metric that they evaluate, which can be EC50, IC50, or KI. And in this way, we retrieve only the experiments that have failed in specific drug protein pairs. Then we use the retrieved data in order to find again the feature vectors that we have previously imported. So this is the eighth step where we proceed to the machine learning pipeline. Here we see how we format the negative class, which represents that drugs that cannot be used in a protein, and the positive class, which are the drugs that are used in a protein. In the ninth step, we split the data in training and external test set. The split is done with a 70% of the whole data set. So we take the 70% of the positive class and the 7% of the negative class in order to design the training data, while the 30% remains for the test set. The data are random sampled. So the final step of the pipeline is to train our machine learning algorithms, which is, in this case, our random forest, support vector machine or HVM, k nearest neighbor, or as it's known as KNI. So we train those three machine learning algorithms using k-fold cross-validation. What we have achieved with this pipeline, as we can see here, we have used two types of node embeddings and we have performed the same type of analysis. So for the node to vec, we can see that we get a very good performance for both all the three classifiers. However, the HVM managed to outperform the other two. Yet we can see that in both 10 cross-fold cross validation and test set, we have the algorithms fails in order to predict totally the negative class, which is the drugs that do not that cannot be used in a protein, while the sensitivity is still very low. While when we perform the same pipeline with the fast-RP node embedding, we can see that we have a 10% increase in our accuracy, which is managed to achieve a 96% performance and an equivalent performance in the test set, which indicates the generalization ability of our models, while the sensitivity and specificity in both 10-fold cross-validation and test set are in equivalent amount. These results indicate the ability of graph machine learning to be applied in a knowledge graph for drug repurposing. So this pipeline has been applied in a knowledge graph and has produced accurate and robust predictions. Those predictions are integrated with the docking results, providing new targets for existing drugs. Based on this pipeline, we have our first repurposed drug, which is now under experimental evaluation, and our first patent is on the way based on this platform. At this point, I would like to thank my team and colleagues that we have worked so many months on this project. And I would like to thank you for your attention. We are now ready to answer your questions. Thank you very much.
Yeah, thanks a lot for a wonderful session. So we have some questions. Let me point out. So it's a question from the Andrew. How many different kinds of node in the graph?
33. There are 33 different databases, so each of one of those 33 databases has its number of nodes. All the nodes are approximately 300,000.
Okay, thank you. Let me move on to the next question. It's a question from Linda. What do you think makes the graph interesting compared to the other machine learning?
Yes, the difference of this pipeline based to the other machine learning pipelines in the graph machine learning is the fact that we have used the negative class according to the experimental data. Most approaches that currently are employed just take a positive class, the known pairs, and they just mix the data and perform a random sampling. So in our case, we use experimental data as negative class.
Okay, so the next question was from Andrew once again. How do you extract the relation?
All the relations that have been added in the platform were collected from public repositories, were pre-processed, and they were added in Neo4j. And the way that were added was done in order with a simple query to get the relationships. So basically all the relations are from public repositories.
Thank you. So we have 2 more minutes. We'll go with 1 or 2 questions. Let me go to another one. So what do you classify with the models? I'm not familiar with this domain. The question from Jean.
So this we don't do exactly classification, but link prediction. We predict, we classify which drugs will bind to a new protein target and which drugs will not. So if we see the problem as a binary classification, is about linking a drug to a protein and a drug that cannot be linked to a protein. If we have an existing drug that is in the market that can be linked to a new protein, then we achieve drug repurposing.
Okay, so the last question, which one we have it, you have mentioned dot product of Hadmod gate. Isn't that the quantum gate?
It's the Hadamard product. It is like the it's the Hadamard dot product. It is like the dot product, but with not the summing part. It's only the pairwise multiplication, as it was shown in the slides.
Okay, so last question. So yeah, it's from Rohit Kumar. Can you provide the link of full ML algorithm integration with Neo4j?
The algorithm is not provided, but the pipeline can be reproduced according to the Neo4j documentation. If you would like, you can contact us and we can see how we can share the algorithm.
Okay, thanks for the wonderful session and also our time. Thanks to all the viewers. So we have another session. It's related to the knowledge graph. We'll be starting shortly. Yeah, please keep in touch. Yeah, thanks to all. Bye.